Yeah, good afternoon from Germany. Welcome to our webinar, iOS webinar. And my name is Tim Sparwasser. I'm heading the Institute for Medical Microbiology at the University of Mainz. And it's a great pleasure to have Hans-Martin Jäck here, who's giving a talk about passive vaccination. And Hans-Martin is a biochemist who studied in Tübingen, and he did his PhD first at the Max Planck Institute in Tübingen, then he followed his mentor to the famous Basel Institute for Immunology and moved on to UCSF in San Francisco. Became an assistant and associate professor at the University of Chicago. And since 1997, he is actually heading the Division of Molecular Immunology at the University of Erlangen. And I guess I don't have to really introduce him because everyone knows him. He used to be president of our German Society of Immunology. He's an editorial board member of the European Journal of Immunology, and he's probably the B-cell expert. He's heading currently the European study group on B-cells. And that's why we are very pleased to have a real expert here on B-cells, and he's going to talk mainly about passive vaccination. And I should just remind you, please don't forget to enter your questions, because you have the chance to ask Hans-Martin some questions about his talk at the end of the talk. And we are looking very much forward to your presentation. Thank you very much, Hans-Martin, for giving us this talk.
So thank you very much, Tim. First, I would like to thank Rita Carsetti, who actually heads the educational committee of iOS, who invited me to give a presentation today. And I have to say it's a real pleasure and honor to give today a presentation about my favorite molecule. It's antibodies and the producers of it, the B cells. And I will talk about passive vaccination, which is, as you will see in a couple of slides, actually a very old way to treat infectious diseases and tumors, etc., etc. I've divided my presentation in 4 parts, and I keep about half of it more at the educational level, because I really don't know whether all attendees know about B cells and antibodies. So I do it quite educational, and at the end I will introduce to you some work we did to make monoclonal antibodies, human antibodies, which neutralize the coronavirus 2. So coronavirus, I don't go in the disease. You all know it's a big threat all over in the world. Even in the smallest country, the coronavirus exists. And right now, because we do not have vaccination, we do not have a good therapeutic treatment. Actually, we have 3 major elements to protect ourselves, protect others, and also to reduce transmission. It's social distancing, which is very hard for all of us, especially in science, where you have to communicate a lot. Washing hands, very important, things which was known since 200-300 years, that washing hands reduces infection, and wearing a mask. And you might have, especially in North America, followed all the discussion about wearing masks. I don't go into politics. The only thing I would tell you is, if you are not sticking to these rules, to these 3 elements, when you release the lockdown, then it can happen that you get a spike in infection. And I'm very sorry for all our colleagues in the United States. You see here over the last 2 weeks, because people did not keep social distancing, didn't wear masks, United States had a spike, incredible, especially in the southern states. And I wonder, when Europe, Canada, Japan, China, other countries open their restaurants, schools, etc., and don't keep the mask on, what will happen then? I hope nothing. We'll see. So therefore, I guess we all of us everyone in the world, yeah, even people who don't understand immunology, wait for a vaccine. Yeah. So I guess this is the most important thing. And I guess most of the research, a lot of research, and a gigantic amount of money is going right now into vaccine research and bring this as soon as possible, yeah, into to the people, which I completely understand. And I, as a big hero of vaccination, completely support. So my talk is about passive immunization. It's something different. You transfer already weapons, the antibodies, which react very fast. But I will give you a brief overview about vaccination, where we stand today. So we have basically 5 different approaches which are right now followed in the field. That's a live attenuated virus approach, which is very successful for polio. This is polio, measles, and mumps. Then we have inactivated virus, which we still use to protect ourselves against polio. Works very well. Then you have protein-based vaccines, for example, diphtheria, hepatitis A, B, and virus-like particles, which are empty. They can go into the cell, but they don't have RNA or DNA in it. And then we have vector-based vaccines, as I remember the Ebola vaccine, which is approved, is a vector-based vaccine. And we have a completely new approach, which was never licensed so far, and it was, I guess, never tested in humans before the clinical trials with the RNA virus. It's nucleic acid vaccines. And I just thought I will tell you a little bit about these two. And the first clinical trial, actually, really the first one was by Moderna, who actually uses the RNA vaccine, was done. I guess the first person was injected in Seattle. And it's very clever. It's a very nice method. So you either inject DNA into intramuscular, and you do kind of electroporation, what we do usually in cell culture, or you use RNA packed in a lipid coat and inject it intramuscular. Then the DNA as well as the RNA is taken up. Then the DNA has to be transcribed into mRNA. You get viral proteins. They are secreted by the cell or exposed in the cell, taken up by dendritic cells, and the dendritic cells prime the adaptive immune system, prime naive and T cells and B cells to mount a very good immune response. Then the other one, just to summarize, the other classical one. So you have the replicating viral vectors. That's a measles-based virus for Ebola vaccine. This is licensed. Then we have non-replicating vectors on adenovirus-based, no licensed vaccine yet. This is an approach which is in the press right now, for example, from Oxford, which is relatively far in the trials. Then we have subunits of the coronavirus, which I'll come back later, spike and M protein, and we have virus-like particles. And we all hope that at the end, and here a little bit summary what we have. This was July 1st, 2020, from a very nice article from Ralph Hemke. We have 136 vaccine design trials, preclinical trials, and increasing almost every day. We have 13 vaccines in phase 1, 4 in phase 2, 0 in phase 3 today, but 2 are approved, and one is from Moderna. And unfortunately, we do not have yet a license for clinical use, i.e. that it can go into the public, that doctors can vaccinate people who need the vaccination. And I have summarized here, don't go through this, very nice links where you can follow here the involvement or the improvement and the development of vaccines to make active immunization. Very nice homepage, especially New York Times, I have to say, was very impressive. So and we all hope at the end, and I don't go through this in detail, that vaccination or even infection establishes the holy grail of vaccination. Memory, long-lived memory B cells and T cells, or long-lived plasma cells. That's the wish which we all have, which works very well with the vaccination which are licensed nowadays, like polio or measles. Works very well. We do not know yet whether this works for the vaccines. And there's a very nice review article just published by Robert Samstein, which summarizes state of the art, let's say 2 weeks ago, what we know about interaction of the virus with the immune system, vaccination approaches, also the role of viral proteins in the immune system. So it's a very, very nice summary. I recommend this to everyone. So problems with vaccines. Once we have a vaccine, there's no problem. If it's safe, that's the way to go. We all agree on this. But the first vaccine trials are actually promising. These programs, which are volunteers which have been vaccinated, was not toxic. Most of them, at least I know of, and they have also neutralizing antibodies. But we really do not know, no idea how long, if these volunteers will develop memory B cells, memory T cells, and especially long-lived plasma cells. And if you want to know everything about memory B and long-lived plasma cells, go to the webinar from Andreas Radbruch, which was given about 3 or 4 weeks ago. Achieving global supply for vaccines seems a little bit unrealistic. So if we have the first 10 million vaccines, well, not everyone will get it. And you might have read in newspapers, which I do not support at all, some countries, including my country, already pre-ordered vaccines and prepaid vaccines which are even not yet tested. So it's a incredible race for the vaccine. So poor countries might lose, and wealthy countries might survive. So we'll see how this final layer will solve once we have a vaccine. And we also don't know whether we can vaccinate high-risk populations like elderlies in nursing homes because they might not have a good immune system. We know that as older you get, as worse the vaccination can be. And as long as we don't have a vaccine, global economic downturn will continue until we have a vaccine. So we definitely have to think about therapeutics, development of therapeutics, be it antibodies or be it other ones. And there is a very nice review article. It was written about SARS 2003, the SARS 1 before the SARS 2, and the MERS virus. I'll come back to this later. But the concept is the same. So it's a very nice article which summarizes all the Achilles heels of the virus, where we could attack it by antibodies, and this I will talk about it, and also with small inhibitors. So therefore, I guess we need therapeutics, and one is here this wonderful red molecule, antibodies. So that's one approach, which we can do. Now, passive immunization is very old, and I thought I'd show you a little bit of development, how passive immunization treatment with antibodies developed. The first report of antibodies that something like this exists came from Behring and Nissen in May 1890 in Berlin. They found that serum from animals which were immunized with anthrax killed anthrax in vitro, but not another strain. And serum from non-immunized didn't work. So what they have discovered is inducible, soluble, and specific something in the blood. No idea that it was a protein or antibodies. Then, and that is the reason why I put the months on, in December 1890, and this is something you read in all textbooks, discovery of antibodies is not completely correct. This was the first description of serum therapy to treat disease in an animal model with antibodies. And what Kitasato and Behring did, they immunized a rabbit with weakened tetanus, took the serum from rabbit, transferred it to a mouse, and came back with a pathogenic tetanus, and the mouse survived. It not only worked therapeutically, preventive, it also worked when the mouse was already sick. So when they gave antibodies, the mouse recovered. So very nice. And then 1 year later, and I bring this because this was actually important for the clinical trials, he did exactly the same for diphtheria. A year later, showed that serum from rabbits which were immunized against diphtheria protected, in this case, guinea pigs. And then it took only, and this is very fast, it's maybe like vaccination today, it took only 2 years or 1.5 years to bring this therapy which worked in animals into clinical trials. And this was done by Behring and Paul Ehrlich with 220 patient children who came down with diphtheria. And very important is, this was the plus of Paul Ehrlich. He titered the serum. So he knew that the serum he gave to the children, had protective antibodies in there. And you can see here, if he gave the serum early enough, almost all the children were saved. So this was a gigantic improvement. And you can see here, diphtheria was 5 years later, at least in Vienna, almost no problem anymore. Only 70 children died, and before 1630 died. So this was a big success. Paul Ehrlich already suggested that he can use, and he coined the term antibody. This came from Paul Ehrlich. And he also here on a German bill, which we saw at the mark and now euro, it's Paul Ehrlich, and he said that antibodies also are very good magic bullets because they can recognize, and with the C part, they can eliminate magic bullets against diseases, tumors, metabolic diseases, and inflammation. The next breakthrough, very important, and I'm almost done with my history, came when George Köhler and Cesar Milstein developed a method to immortalize B cells from mice. It also works for human, but works very nicely for mice. Rabbits. Catherine Knight has established this at Loyola University, and in rats. And so you can generate single cells, which grow indefinitely in culture, and all of them produce the same antibody. And that's what we call monoclonal antibody. And they also solved one last mystery, or not the last, one big mystery at this time. They showed that one B cell makes one antibody. This was the final proof of the idea. Today, before Corona, almost the most use of monoclonal antibodies are related to treating tumors and inflammation. You see here a list of different antibodies, and you might remember that Jim Allison and Tasuku Honshu received the Nobel Prize 2 years ago for developing their checkpoint inhibitors, which was a big landmark for tumors. So if I remember, only 1 or 2 antibodies are in the clinics, are licensed to treat actual infectious diseases. Now we come to Corona, the human coronavirus family, and I just want to make a small summary about it. I'm not a virologist, but a little bit I know. We all know about this. The pathogenic human coronavirus family consists of 7 members. We have here one which caused the cold. Some of the colds we come down almost every winter, at least me, and small mild respiratory illnesses, like the HQ1. And then we have also 3 strains which really can cause very severe acute respiratory syndromes, lung diseases. The first one was SARS-CoV, 2003. The next one was MERS, 2012, with a mortality rate, frequency of 30%, but it was somehow maintained in the Middle East. And then finally, the one which we have to do right now, the Corona CoV-2, and maybe it's not correct anymore, I'm sure, is we have about 10 million infections, at least detected infections, and about 500,000 deaths. So that's a big threat, to mankind. Now, coronavirus, is a very interesting virus. It contains a single-stranded RNA, the largest RNA virus, which is discovered so far. It has about 26 kilobases long. It encodes for polypeptides, which are cut by peptidases in smaller proteins. For example, one is the spike protein, which I come back right now. And I only want, and here again, this is the review by Samstein. They also discuss the role of some of these proteins encoded by these open breathing frames in regard to manipulating the immune system. So read this; it's really a very, very nicely written paper, not only for immunity, also for function of viral proteins in the immune system. Now come back to the proteins, which I guess we are interested in looking for a target. We have a nucleocapsid protein, which is the protein which actually covers the RNA and builds the nucleocapsid inside the viral particle. And the viral particle, I forgot, it's a membrane virus. So it takes the membrane from the host cell with it, so like HIV. We have a membrane protein M. It is important to organize the assembly of CoV-2, and envelope E interacts with the M to form the envelope. And then the one which I would like to stick to is the spike protein, because the spike protein is on these green, big bubbles on the surface, shown in the file. They are actually very important, are required for the entry of the virus into the cell. Without spike, no infection. And here, very schematic overview. Here is a schematic primary sequence of the spike. It has two functional regions, a S domain and the S1, S2 domain. The S1 domain contains the binding site, which is called receptor binding domain, which interacts with the host receptor, which I come back here in a moment. It also, S2 is important for fusion. It contains cleavage sites, which the protein has to be cleaved, that a fusion peptide is released, which then allows the fusion of the viral membrane with the host membrane, which then releases the RNA into the cytoplasm. And the spike protein, it's a large, gigantic complex. One molecule is about, one single molecule is about 130, 140 kilodalton long. It's highly glycosylated, it's about 20% carbohydrates on it, and it forms a trimeric molecule on the surface. And this, and this spike actually interacts, I've labeled this here green, with with angiotensin converting enzyme 2, not only, but found on epithelial cells, for example, in the lung and also in the gut. So that's the entry receptor for the spike into our cells. And then you get a membrane fusion and the RNA is released and translation starts. Now this ACE2 is a very interesting molecule, and I read this all up after the corona came out. I didn't know this before. ACE is an enzyme which converts angiotensin, already a proteolytic form of AT2, into a smaller AT protein. And this binds to the mast cell receptor, which actually inhibits inflammation fibrosis. So signals through ACE2 are important for epithelial cells because it's lung protective. So if the virus binds to ACE2, it cannot do its function anymore, very likely. And if you use, for example, soluble ACE2, you might also catch out the AT2. So it's better not to mess around with soluble ACE2 or shed the ACE2 from the surface. So I guess in our opinion, it's better to use antibodies to block immediately the binding of the virus to the ACE2, that the ACE2 still can do its function in lung protection. But that's just a hypothesis. So how do you get antibodies out of, how do you generate human neutralizing antibodies? Basically, there are two major approaches, several others, but I focus on two major approaches. The first is you take blood from from COVID-19 patients which have recovered. And then you either produce serum from it, and you directly transfer the serum to other patients. And that's a classical serum therapy after Behring and Kitasato. You produce everything which is in there, maybe not only the antibodies. Or when you isolate the antibodies, you have a polyclonal serum. Or you isolate plasmablasts, from the blood, which are present during an infection. And then you do single cell sequencing. You isolate the variable regions, you clone it into a vector, you isolate the antibodies, and you test and screen for binding and neutralization. And then you have a monoclonal antibody. And this monoclonal antibody you can has a defined specificity. You can test the toxicity beforehand, and then it's, in our opinion, safer than if you use total serum. The other approach is to use human antibody mice, and I will come back to this mouse which we have developed in Erlangen. There are a couple of companies who made different mice. So the mouse has an entire human repertoire, makes only human antibodies. And so the best would be you infect the mouse, but this is impossible right now because mice cannot be infected with corona-2 because ACE2 from mouse doesn't bind to CoV-2. And transgenic mouse models, which have the human ACE2 are not yet available, at least to us, commercially, yes. Or you immunize with spike protein, and this is what we actually did, and I come back to this right now. So I cannot go through this part, yeah, but I have again summarized 3 nice review articles, yeah, which actually summarize, yeah, the state of the art with the serum therapy, and also 2 nice antibodies, which at least, yeah, update, yeah, 4 weeks ago, what kind of antibodies, yeah, we already have made. Most of them are from COVID patients. Good. So monoclonal antibodies. We think if we don't have a vaccine yet, yeah, monoclonal antibodies are very good because they act immediately. You give it, yeah, they are there, yeah, in large amounts, and they can neutralize the antibodies. So they should do their time within minutes, like the innate immune system. You also have a fast readout to test for efficacy in clinical trials, because you give the antibody to COVID patients and you look, yeah, whether you can reduce the hospital stay, whether you reduce the viral load, and yeah, whether you reduce the numbers of diseased patients. So these are the readouts which can be done in a couple of weeks, not months or years like for antibodies. And you can pre-screen for something which is very odd, a syndrome called antibody-dependent enhancement of disease. And what is this? This is something, yeah, where antibodies do exactly the opposite what they should do. They are, if antibodies are already there, or are given and you have a viral disease, the virus is around, antibody binds to the virus, here see left upper corner on A, and it enhances actually the virus. So the virus, yeah, can infect better and you get worse with the antibody. And this is called antibody-mediated enhancement of disease. This was found, yeah, in a very old measles vaccine, yeah, years ago, before the safe measles was activated. They also found it in dengue, immunizations with dengue virus, and it was reported, yeah, for SARS-CoV, MERS-CoV, and RSV. But mainly when you transfer serum, yeah, from patients, yeah, or from mice into mice, or if you infect them. So this was a problem. There are some antibodies, and it's not clear why some antibodies do it and why not, yeah, actually enhance your viral infection. But this can be very easily screened, yeah, in animal models, yeah, for example, in non-primate monkeys, or in mice, yeah, for coronavirus, which have the human AC. So you can easily test, yeah, whether your monoclonal antibody, yeah, does this. And you can also modify the concentration of the antibody, that it doesn't bind to Fc receptors and it doesn't activate complement. So you can reduce or abolish, yeah, this phenomenon completely. So we thought, in contrast to serum therapies, monoclonals are safer because they have a defined specificity and can be pre-screened for side effects in animal models. So we decided to go for this part here, yeah, for the gray part, to go for the mouse, because as I already mentioned, yeah, about a couple of years ago, we have developed a human antibody mouse, yeah, the so-called Triani mouse, with a very small team at this time, Nigel Carlin and Matthias Wabel, a small company, a Triani, a company in San Francisco, and 3 people in my lab, Edith Roth, Jürgen Wittmann, and Miriam Metzner. So developed a mouse which has actually here the entire human antibody. And this we used, yeah, so we immunized the mouse, and we took the spleen and made hapedomas. We also work on this part, that we can actually isolate plasmablasts from this mouse, yeah, to go into this route. It might be faster than hapedomas. Katrina Pracht is developing right now a method to isolate plasmablasts or memory B cells from the mouse. So if I say we, yeah, it's a team, yeah, who is working on these antibodies. It's called the COVER-IB team for Coronavirus RNA Antibody Consortium. And it consists of the lab of Klaus Überla, Thomas Winkler, and my lab. Klaus is the virologist, Thomas and I, we are the B-cell immunologists. And this is the team. I mention all these people later on, yeah, what they do in our work. And Nina Gleusner and Thomas Winkler were very helpful to convert our CMI mouse antibodies, yeah, into human antibodies and make recombinant human antibodies, yeah, in cell culture. So first, what we did is, yeah, we immunized our Triani mouse with a DNA plasmid, yeah, by intramuscular electroporation. So it's with the DNA vaccination. And we came back with isolated spike protein and adjuvants, did a reboost, yeah, and took serum and looked whether we have antibodies. And we used a flow assay, which was developed by Sophia Peter in Klaus Überler's lab. It was a very clever thing what she developed. She used HEK cells, transfected the HEK cells with a GFP reporter as a transfection marker, and then she put the spike in either of Corona 2 or of Corona 1, and I call the 2003 now Corona 1. And then we stained it, we added serum from the mouse, and we added the antibody which detects the serum. And then we gave it on the green cells, the GFP positive cells, and you see the result, very impressive result of the serum analysis of the mice. You see the serum of the Triani mouse bound very nicely cells which express the spike of Corona 2, but not from Corona 1. So apparently the serum was already specific. And here as a control, we used the antibody which Thomas Winkler isolated from a COVID patient, which binds very nicely, but unfortunately didn't neutralize. But it's a very nice binder and one of our controls. Just as a side visit, Wolfgang Schuh, Mayola Hauke, and Edith Roth have developed a flow assay exactly on Sophia Peter's basis to detect antibodies IgM and IgE and A in one staining from COVID-infected patients. This is a non-infected person, and as an infected person, you see, I can nicely see the IgG and the IgM. And it always went along with all the ELISAs, we did in controls, and sometimes it was even more sensitive than the ELISA, which we used for serum antibodies. So it's a very nice assay. Now come back to our hybridoma project. So what we did is, Edith then did, after the last boost took the spleen, did a classical PEG fusion, and got hybridomas. This is actually the first hybridoma I looked at it when I saw it and took it with my iPhone. You see a very nice growth of hybridoma cells. It's always very nice to see this. We don't know whether they are positive yet, but anyway. Then we used exactly the same assay to look in supernatants for binding of to the spike. We did this and we got about 35 binders, and then we used a ELISA assay to test, at least pre-test, what part of the spike protein is recognized by our antibodies. And this was developed by Sebastian Schulz, Tobit Steinmetz, and Dirk Mielens. They put RBD, this is the domain which binds the host receptor, or the S1 domain, and we repeated the same now with the S2 domain. On the plate, we put our hybridoma supernatant on it. We looked at whether it binds. And you see here 3 hybridomas which nicely bound to RBD, bound to S1, but most of them did not bind RBD, but they bound S1, and some of them here which didn't bind S1 bound S2. So we got a whole variety of different antibodies. Then we did a pre-test for blocking. Do they block? And Tobit Steinmetz and Edith and Sophia and Manola developed 2 different assays to look for blocking, a ELISA assay and a competition assay. For example, we used again our 293 cell lines with the spike. We added soluble ACE, which was labeled, fluorescence labeled, and we added increasing amounts of our antibodies. And we called them DRESS antibodies for Triani Erlangen spike antibodies. So all the antibodies are called DRESS. And doing this, I didn't show you the original data, is we found that all the 3 antibodies which bound the binding domain, the other ones didn't compete with soluble ACE with the EC50 value in the picomolar range, which is a very, very good blocker in both assays, in ELISA and in the flow assay. And then, of course, the most important, do the antibodies neutralize? And there, Klaus Überler and Sophia Peter, under S3 conditions, to work with infectious virus. So this was a isolate, from a patient, which was diagnosed in Munich. And Klaus got the virus and he cultures the virus. So you put the virus onto virus cells, you add increasing amounts of antibody, and then you detect viral RNA with qPCR. And then you can determine, over the concentration or over a titration curve, you can determine the IC50 value. So the amount you need of antibody you need to neutralize 50% of the antibodies. And we were in a range of nanomolar range, which was very good when you compare all other publications from COVID patients. And we were very fortunate, because Tim said that I was at University of Chicago. Not completely correct. It was at Loyola University of Chicago. That I remembered a very good friend of mine and my former chair at Loyola, Tom Gallagher and Catherine Knight. So I called them up and said, yeah, we have a different test. We have a pseudotype neutralization assay. This can be done under S2 conditions, and Tom is an expert in coronavirus since I know him, since 25 years, before all this corona came up. And we sent him our antibodies, and he also tested our antibodies against VSV, SARS-CoV and MERS-CoV, and he found that our antibodies only blocked CoV-2 almost with the identical IC50 value as Klaus found in his neutralization assay with the infectious virus. So this was a very big thing. And Tom, if you are there, I miswrote your last name. Thank you very much again. This was a very important piece of data. So just to summarize what we have found, we have done so far 2 fusions. We screened 3,500 hybridomas. All the screening was done by Edith and Sophia. So the 2 did this at night over the weekend. Was great work. Thank you so much. We identified 35 spike binders in our flow test. 3 of them bound to S2 in ELISA, what was found by Tobit. 3 of them bound spike. They did not bind to RBD. They did not bind to S1, and they did not bind to S2 in ELISA. But they neutralized. Weaker than the ones which I come back now, but they neutralized. And then we found 6 RBD binders by ELISA. 2 didn't neutralize. So this was very interesting. We have 2 antibodies which bind to the RBD, but it doesn't neutralize. It's relatively large, so it could be that it's outside of the contact point of AC. And we find 4 very strong neutralizers which bound to the RBD and neutralized in both assays, pseudo and the regular viral assay, in a low nanomolar range, which is very competitive to almost all antibodies which are around. So ongoing research is, of course, the next one is we have to do proof of concept in an animal model, and we are very fortunate to have 2 great collaborators who stand by to receive our antibodies in milligram amounts in non-human primates. That's Christine Stahl-Henning from the Primate Center in Göttingen. And then Thomas Grunwald will establish a mouse line which expresses the transgenic mouse ACE at the Fraunhofer in Leipzig, so then the mice can be affected. We also do epitope mapping right now with X-ray and cryo-EM. Affinity constant and recombinant antibody is done. This was done by Thomas Winkler, and we have a very nice setup to make at least antibodies for the first tests in mice. So I would like to finish with my presentation with the team again. It's the 3 teams. It's my lab, Klaus Überler and Thomas Winkler, and the people, the colleagues in our lab who has contributed to this program project. And I have to say, compliment to all the people, all the students and the technicians who actually worked overtime and over the weekends. This was really amazing and reminded me of my PhD times and postdoc times at UCSF. Thank you very much to all of you, and thank you to the audience. And I'm more than happy to answer questions if I can. Thank you very much.
Thank you very much, Hans Martin, for this excellent talk. And I see a lot of questions here already. There are some questions about the general part about the COVID-19 pandemic. But I think we should first jump into the more specific questions and then maybe go back if we still have time. So one question which actually popped up several times was the question with regard to the decline in antibody titers in patients who recover after 2 to 3 months. So what does this mean for the memory response and for the success of a vaccination approach?
So yes, right. There are a couple of publications and more are coming up every day, to look in longitudinal studies for patients who had COVID very strong, mild or asymptomatic, because asymptomatic you can get them if they are positive and they don't have any disease at all. Antibodies decline in most of them, IgG in IgA, yeah. So this means, yeah, that usually there are that either the one cyclic infection did not generate, yeah, long-lived plasma cells. Because to keep, yeah, antibody, yeah, levels, yeah, up, yeah, you have to have long-lived plasma cells. However, I'm not so surprised, yeah, about this, because when we immunize mice once, yeah, and with alum, a very strong immunogen, and we look, yeah, we also find, yeah, that and IgG goes down, yeah. Now we should boost, but the patients were not boosted yet, yeah. Then you decline, you have the plasma cells go down. You might still have plasma cells, which make relatively small amount of antibodies, which might be enough, yeah, for the first defense against viruses. And the memory B cells, we don't find at all, because you only find them when you re-challenge the patient. So you have to wait until the patient gets infected, or if the patient gets vaccinated. Then if you have memory B cells, then you should find a faster, yeah, increase, yeah, in IgG and IgA, and maybe also IgM, also memory B cells for IgM. So we have to look in these longitudinal studies, yeah, and I don't know if it's published, yeah, because sometimes I lose a bit the overview. You should look for memory B cells. In humans, you can do it. We have relatively good markers, yeah, but we need good staining to look for B cell receptors which bind, for example, the spike protein. And then we would have an idea if you find memory B cells there. So I'm still optimistic.
I think your talk was very convincing. The first questions come already, how people could join you as a PhD student.
Anytime, for good students, we always have a space.
The other question was, so do you, or I mean, obviously people do, but do you specifically look at memory T cell responses as well, especially in the patients which have no symptoms or very mild disease?
You know, we just hold the DFG grant, yeah, Thomas Winkler and I, yeah, and Klaus Überler, and we thought, yeah, to include in our topic the memory T cells, but I thought we completely stay out of it. Yeah, I don't have experience in there. Neither of us have, and in Germany there are very, very good groups, including your groups, yeah, and a group in Berlin who have really set up already a very nice staining with soluble MHC with peptide. I forgot what the method is. They are around the Drosten group in the Charité, and they have all the tools to look actually for T cells responsive, yeah, to a coronavirus. No, we are not looking for this.
Okay. And what about the patients which have no symptoms and also don't show a seroconversion? So what do you think about those cases? And another question that comes here is, what about the IgA response? Do people have high affinity neutralizing IgA?
Mm-hmm. So to the first one is, I don't know what people speculate in the literature. I just can speculate. One simple explanation is, yeah, the PCR test was wrong. So I don't know whether these asymptomatic people which are published, and it's very hard to find out, especially when you look to this pre-publication server, which I never do. Yeah, you put everything on there. So I cannot evaluate, never went through a peer review. I don't know if these asymptomatic people were continuously tested again, yeah, whether they are really virus positive in a second or third screen. No idea. Might be false positives, because you get this. If the lab is not careful, depending on the lab, it's easily contamination are possible. Yeah, regarding to Klaus Überler, who is also runs the diagnostic lab in Erlangen. Yeah. But let's say there is coronavirus there, you don't have a response. It could be that you have neutralizing antibodies from a cross-reacting antibodies, and I don't know if people looked in these non-symptomatic people, if they have antibodies against SARS. MERS is very unlikely, except they have been in Arabia, or against these cold viruses, which we are challenged every year. It might be a epitope which we might recognize, which might cross-react, and this is why they have a protection, and don't make antibodies at all. You catch it before you activate the B cells, like with the rhesus therapy for histoincompatibility. But that's just a speculation. Yeah. IgA, very interesting. I followed this up. usually when you infect through the mucosa, the first antibody, what you find is the virus goes in the epithelial, it goes in there, it finds prior patches, you find IgA, you find high-affinity IgA, but this is actually the antibodies which decline relatively fast, faster than IgG. So the second one is the virus makes it to other lymphatic organs, and then the main response is IgG.
You focus in your talk mainly on the antibodies against the receptor binding domain, the neutralizing antibodies. So what about the ones against the repeat-2 domain, which blocks fusion? Are there synergistic effects as well? Can you imagine?
We don't know. I mean, we don't have this antibody. There is a Chinese group published a antibody, neutralizing antibody. It neutralizes 10 times worse than our antibody. It doesn't bind to the RBD. It binds somewhere, I guess, S2 or S1. It blocks, it could be a conformational change, it could be, and it's a very attractive idea. The corona has a second receptor, and the corona is heavily glycosylated, so it might be that you have a second receptor, and this antibody blocks, the second receptor, so the virus still infects, but not as efficient as without the antibodies. There are antibodies which actually help each other. Regeneron, this is our competitor. It's a little bit hard to say. It's a company with 1,000 people. They also have a human antibody mouse. Actually, the first one was a complete kappa and heavy chain repertoire. And they just published a Nature paper with about 20 monoclonal antibodies. And they have antibodies which bind to the RBD. One neutralizes, the other one doesn't. But when you put both together, it neutralizes better. So there is a cooperativity between two antibodies. We don't have such antibodies yet, but they exist.
Okay. Something which also puzzles me a lot because I simply don't understand it, but I'm not a B-cell person. So the high titers of anti-S and anti-N, IgM and IgG correlates with the worst clinical outcome. So what does this mean? Or
I should know the answer.
Or could this somehow be an overshooting immune response being reflected also on the B-cell side?
Well, I do not know whether really very, very severe symptoms are always correlated with a high virus load. It also might be that these patients have a low ACE amount, so less protection of the lung epithelial. There may be mutation in ACE, which is all beginning to, which bind the virus better, it's possible that, and then let's say, if you have then a severe immune response, it could be that you have a enhancing immune response, help adjuvant effect on the B-cell compartment. That's really a speculation, and I don't know if other people have better ideas. And I didn't read this article completely yet, which I cited. Maybe it's an idea to follow up, but I don't have a good answer. I admit.
So, and if you talk about antibody-dependent enhancement, what are the factors which determine it, and what can you do to control it?
We really don't know. It's not known. Everything, but how we can control it is first, and this is what we do actually in our animal model with the non-primate apes. We give the antibody to the apes. It's also, thank God, it's all approved, and we give the virus. And then the antibody should neutralize, but we also look for pathology in the ape and hope that the antibody does not enhance the virus. So we will test this in the animal model, at least in the apes. And then, of course, you have to go in phase 1 and look whether it does it too. Once you know it does, then you can modify the FC region. And if you remember, there are these two possible mechanisms. It binds to FC and more virus comes into macrophages, or more virus comes into cells which have a complement receptor. Okay, and if you block the complement and the FC receptor binding site, which you do not need for neutralization, then you might succeed. But we can all test this in animal models. And once the mouse is there, then we can do it in a mouse easier than in the ape. And with the serum you can't. And vaccination is a problem. if you vaccinate, you might get antibodies, which exactly do this. So each of the trial can happen. What happens to the measles vaccine, the mumps, was a mistake. Very early, the first mumps vaccination were actually deleterious, You vaccinate, and then they got mumps and they got it even worse. Not deleterious, they got it worse. But I'm not aware. I read a couple of review articles in the original. There is no really structure on an antibody which determines that this antibody might be IDE.
Okay. So this answered also one of the questions if you're doing studies in non-human primates, which you just answered. One question that, of course, everyone has, and I guess this is also pure speculation, but maybe you could comment on your monoclonal. When do you think a vaccine or an antibody might be available, and what are the strategies used to accelerate this process? You alluded to.
I didn't understand the last one. What was can you repeat?
The strategies to accelerate this process.
Oh. Well, we are ready to go. We have the clinical partner set up. We have 3 hospitals set up who would do the trials for almost nothing. We have a very good company in California who would make us the antibody in 4 months. Well, you know what we need? A little bit money. So therefore, I'm a little bit disappointed about our government in Germany that they spent 250 million only exclusively in vaccine research and nothing in therapeutics. But we need a little bit money. And then I guess we can go into clinical trials always times 2 in 6 months.
So I think you advertised nicely your strategy, and maybe a sponsor is listening. Another question that many...
We are not alone. There are a couple of other people also have good antibodies. You're not, but it's about this one. If you have 20 antibodies around, you can never produce antibodies for the whole world. So even if 30 antibodies around, it's no problem. Everyone produces it locally and supplies the people, and then it's fine.
And so many people are asking the question, so what are the adverse effects of your monoclonals and what's the safety profile? Well, the adverse effect,
antibody therapy is incredible. If you test it right in animals, very safe. All the antibodies against tumors, I showed you in this one slide. No side effects. From the antibody alone. So all phase 1 trials. There are no side effects. And the only side effect I think is this antibody dependent enhancement of disease, what I outlined. But this we can test in non-primate apes and in mice. Okay. And once we have this, we have to do phase 1 toxicity studies, clear, and we know whether it's safe. But because many, and even serum, you get. If you have a snake bite, you don't get monoclonal antibodies, you get a serum, I don't know, from horse or whatever, and it's also fine, So antibodies are relatively safe therapeutic, if they only see the coronavirus.
And if you compare the efficacy to, let's say, a normal standard active vaccination, would you dare to make a comparison? So that's one question.
No, vaccination is always, a good vaccination, you don't need antibodies anymore. Vaccination is the thing we all should go for. Polio is done. Measles is done. Mumps is done. We don't have any antibodies against measles. We need antibodies against tetanus because there are crazy people around in this earth ball, who don't get a tetanus vaccination. But usually, once you have a vaccination, antibodies are obsolete. Except for people who cannot be vaccinated.
Of course. And what's your opinion about this, the mutating virus, now these new virus strains which have been described in Beijing, which seem to be more contagious but potentially less pathogenic, and how does this concern the vaccination approaches and your monoclonals, of course, also?
Well, the monoclonals we can easily test. As soon the first couple of mutations which it's published, we put this into our spike. So we put the mutations in. We test in these whether we at least bind and block. We don't have a recombinant virus yet. So this we can test as monoclonals. The vaccine, no idea. You have to wait. And the mutations, there was a very interesting podcast from Professor Drosten. He's the corona hero in Germany. Some of you even outside of Germany. Yeah, he's Dr. Fauci, yeah, of Germany a little bit. He has a very interesting hypothesis, and some other about this too. If a virus mutates and gets more pathogenic, yeah, you might be actually good, yeah, because people get earlier sick, they don't go out of the house, yeah, they don't spread the virus, yeah, and we can actually, yeah, go down in the pandemic. This is one scenario. We have to see how it goes. So mutations are not always, oh my God, yeah, something happens. You have to see. And the first, so the vaccination, this I don't know. The vaccine, I really don't know. Yeah, if this affects the vaccination, if the antibodies produced in a vaccine, yeah, also neutralize the virus, has to be seen.
And could you consider like some competitive inhibitory effects between pre-existing antibodies against SARS-CoV-2 or other coronaviruses together with your therapeutic antibody? Or
no, I don't think so. If there are pre-existing antibodies there, not from, you mean from corona, possible? No, because if the it's even good. If they have pre-existing antibodies against spike, you have the virus which is already a little bit coded with antibodies. So you need less antibodies for the patients. It is cheaper. Yeah, I mean you don't want to mess around with a titer once you have one, but no, I don't see any disadvantages I can think of right now.
Great. So I think we have like some 3 minutes left, which brings me to the more general questions, where I guess also there might be also a lot of speculation here, because these are also questions I really have to say, I would say are open questions. What's your personal take on trained immunity? Like in how far could BCG help, or oral polio vaccine to induce some innate type immune response, which could be protective or not?
Well, sometimes immunologists are a bit ignorant and arrogant people, and maybe I'm also part of it. I had a talk, about 4 or 5 years ago at the workshop, in Africa about innate trained immunity. I thought, what are they talking about? That's stupid. Yeah. Then I read all the literature. So I really read into literature, and it's really very interesting. BCG in African, especially in African, for unknown reasons, I mean, no, we know some reasons now. You are also more protected against tetanus and diphtheria, for example. And also polio, polio apparently, but the live vaccine, people get live polio vaccine also have lower incidence of getting other infectious diseases without being vaccinated. Very, very interesting. We know a little bit how it works. You have macrophages, they take up the virus, and these little things, can really develop into a memory phenotype. It's not that they are memory cell, they are epigenetically imprinted that they produce, more cytokines. So they are there and produce more effector cytokines. They are there and help, when another infection come to fight against it. So I became not a hero, but I became of someone who really should consider trained immunity in any kind of diseases. The BCG trial was negative, was done in Australia, and now they do one with polio. We'll see what the polio says. People who got vaccinated against polio, do they have a lower incidence of coronavirus infection? If yes, well, can it play a role in Germany, or because we are all vaccinated, but we all got the dead polio vaccine, not the live vaccine. So yes, question is yes, we should follow it up. Very interesting thing, not anymore hoax. There's all experimental data and also mechanisms which can explain this trained immunity. Very interesting thing.
So we stopped vaccinating against BCG many years ago. I think I still have the BCG vaccine. So somebody in Argentina wants to know, what is the explanation that we have such a low number of fatal cases in Germany? What's your opinion about this?
Oh, well, at the beginning it started very low, and I thought, oh my God, 0.5%, 0.7%, But at the beginning it was that most of the people got infected are younger people, because they came from Austria. This was the Ischgl, this was the major hotspot which actually infected the southern part of Germany, and there it started, And then it got into older people, and we are now basically at a death rate which is definitely below Sweden, and I guess Well, one idea is our medical healthcare system is just very wonderful. We have a lot of IC units, yeah. The doctors are trained, yeah, to take care about these people, yeah, and surprisingly, yeah, our neighbors like UK and France, yeah, have much less ICU units like Germany. This might it be. And the second is, Germans are very, if they like to follow rules at the beginning until they are sick of it, like now. But we might just have kept, yeah, better the distancing and wearing masks, yeah, than other countries. That's a speculation. But the healthcare system, yeah, is definitely one explanation.
So I think we are almost run out of time, and that's a perfect last sentence. I should also add that Germany is a good place to do a PhD or a postdoc. So Hans Martin obviously still takes application, and I think you gave a wonderful presentation. Thank you so much for this.
You're welcome.
Thanks to the audience for the excellent questions. I still have a couple here listed up, but I'm afraid we have to stop here. And the presentation will be available online within the next 1 to 3 days, I guess. Yeah, it's a pity we have to stop here. We could continue discussing for hours, I guess. But it was wonderful to listen to you. Thank you so much, Hans Martin.
Thank you, Tim, and thank you to the audience. Thank you so much. Yeah. Bye-bye and stay healthy, everyone. Bye-bye.
Bye-bye.